Hey guys, I'm sorry if you guys can hear this heater or one of my two heaters because it is so cold in this building right now that I am a popsicle, but I'm trying to push through so I can record this video for your lesson. Okay, so let's get into it. So today we are starting a new unit. So this is the first lesson of that unit, and essentially this unit is about protein synthesis. So protein is one of the biomolecules that we talked about. They do lots of different cellular jobs, and then synthesis, to synthesize something, means to create it. So we're going to talk about how proteins, how these things that actually do cellular jobs, are created. Okay, and that process can be called protein synthesis, can be called central dogma. Transcription, translation, we'll have a lot of words today. So we're going to go over the overview of it, and then tomorrow or the next lesson, we're going to actually break it down. You're going to have some practice and a quiz over it. Okay, so let's get into it. So today's lesson objective is that students should be able to describe how DNA actually codes for traits in organisms. And we also should be able to define key terms in relation to central dogma. And we should be able to differentiate, so know the differences between transcription, translation, and DNA replication. Okay, and so let me move my face really quick. This right here is a DNA molecule that's untwisted. This is what it looks like when it's twisted, and this is the typical way that you're used to seeing it done. You're used to seeing DNA like that, but just know that all of these are DNA just at different stages, and we're gonna talk about DNA a lot in this spring. Well, it's not quite spring semester yet, but we will talk about it a lot. Okay, so questions that need answers. How exactly does DNA give us our physical traits? So obviously, we all know, the function of DNA is to give us our traits, right? So some people, like Michael Jordan, has DNA that makes him really, really tall and athletic. While someone else might have DNA that makes their vocal cords really, really smooth and nice, and they're able to sing, right? We also know that some people have DNA that gives them brown eyes, while other people have DNA that gives them blue eyes. Right? Of course, we know that. So we already know that DNA gives us our traits, but we haven't really talked about how exactly that happens. How does it go from gene to trait? So we're going to talk about that. Sorry if I'm sniffling, it's gross. I'm sorry. Central dogma is the law of life. So all living things go through this process that's called central dogma. So DNA looks like this, right? We recognize DNA because it has the two different strands. We know that that picture is DNA. DNA is located in the nucleus. Then DNA is converted into what's called RNA, ribonucleic acid, that is found in the cytoplasm. That RNA is then used to create proteins. Proteins are made on protein factories called ribosomes. And those proteins are actually the things in our cells that do jobs. Okay, so they may produce a pigment that makes your hair color brown. It may produce something that makes your bones grow really long so you're tall. Okay, so proteins do cellular jobs. Those cellular jobs are essentially what gives us our traits, our differences from person to person, animal to animal, bacteria to bacteria, whatever. Okay, so central dogma is that thing that explains exactly how DNA codes for our physical traits. This chain of events, so DNA to RNA to protein to traits, occurs in all living things. So, do bacteria have central dogma? Yes. Do humans have central dogma? Yes. Do trees and other plants experience central dogma? Yes, they all do. We all have to have proteins. We all have to have DNA, RNA, all of that. Okay. So everything that is living experiences this thing of central dogma. It is the law of life. Okay. So this law of life, central dogma, is literally, again, DNA is going to code for RNA. RNA is going to code for proteins. Proteins are going to code for our traits. So in today's lesson, we're going to break it down into a couple different parts. So we're going to talk about DNA, RNA, proteins, and then we're going to review central dogma. So let's talk about DNA. So our warm up. What are some fast facts that you probably already know about DNA? Okay, so you might have learned, or you might have told me, you should have learned all of these things, that one, all living things, so prokaryotes, which are simple, small organisms without a nucleus, and eukaryotes, which are large, complex organisms that have a nucleus, they all have DNA. Two, physical traits. So DNA is going to code for our proteins, and proteins are going to give us our physical traits. The third thing we should know about DNA. DNA is a nucleic acid. It's It's one of the 4 types of biomolecules we talked about right before break. We talked about proteins, lipids, carbohydrates, and nucleic acids. Those are the 4 biomolecules. Okay. And we also learned, and when we talked about our evolution and taxonomy unit, we learned that the more similar two organisms' DNA sequences are to each other, the more likely that they have a recent common ancestor. So, some more facts about DNA. DNA is double-stranded, so it has those 2 strands, while RNA is not. DNA's 2 strands are going to complement each other. Okay, so they're going to complement each other. And you kind of can get a hint at what complement means, because if you look at this, the A and the T fit together perfectly. But if you were to try and put that A piece inside of the C, or that's a G, it wouldn't fit, right? So A and T are actually going to fit perfectly together. C and G are going to fit perfectly together. The T and G will never fit together. A and C will never fit together, okay? The only way that they work is if A pairs with T and C pairs with G. They complement each other. Okay, this whole rule of complementarity actually allows us to do a lot of different things. So if I am told that if I am told the sequence, which sequence just means the order of one strand of DNA, all I need is one strand. Because if I know if there's A, G, T, G here, then on the other side where there's an A at, there's got to be a T. They complement each other. They work together. Okay. If I'm told on this side there's a G, on the other side I automatically know there has to be a C there. So if I'm told one side of the DNA molecule, I can easily, quickly figure out the other side of it in order. Okay. So complement, again, is the base pairing rules. So A pairs with T and C pairs with G. I'll go ahead and say the names. Adenine pairs with thymine. Cytosine pairs with guanine. Okay, most of the time you're not going to see those words. You're just going to see A, T, C, and G. But just in case if you see adenine, you're like, what is that? That's just what the A means. Okay, that's the A word. And here they are spelled out if you need help spelling them. An easy way to remember the complementary pairing rules in DNA is by remembering this little rhyme. Apples in the tree. So adenine pairs with thymine because apples go on trees. And cars go in garages. Cytosine pairs with guanine. So A and T pair up, C and G pair up. So now we're going to do some practice. Practice finding the complementary strand to this DNA sequence. Okay, remember, apples go on trees and cars go in the garage. A pairs with T and C pairs with G. So let's do this first one together. If I have an A on this strand, what is going to be here on the next strand, on the complementary strand? Well, apples go on trees, so it pairs with a T. If I have a T on this strand, what goes on this other strand right here? What's the complement of T? Again, apples go on trees. How about C? What pairs up with C? G, cars in the garage. Okay, let's keep going. What about this G right here? G in one strand, its complement is C. Okay, let's try and do the rest on our own. Apples on trees, cars in the garage. Apples on trees and cars in the garage. Good job. So again, apples pair with trees and cars with the garages. That's the only way that they pair in DNA. That's the only way. A always complements with T and C always complements with G. Apples with trees and cars with the garage. Okay, Mr. Burnett asked Zaria and Jesus to write the complementary basis for the following strand of DNA. So this is the strand of DNA. Jesus thinks that the basis on the complement should be that, while Zaria thinks that the basis should be this. Who is correct? Okay, so for those of you guys who said Zaria is correct, you are correct. But now I want you to tell me, why is Zaria correct? What exactly did Jesus do wrong? So Jesus thinks that the basis should be, but that's the exact same thing as that. So Jesus's error was that he did not use the complementary base pairing rules. You have to use the complementary base pairing rules. Okay? While Zarya did use the complementary base pairing rules, where apples go with the tree and cars go with the garage. Excellent. So we already know the DNA codes for traits. So again, let's get a little bit deeper. Let's look exactly how that happens. So DNA is deoxyribonucleic acid. Okay? DNA, deoxyribonucleic acid. It is a nucleic acid, which we said was one of our 4 types of biomolecules. So whenever we talk about biomolecules, we learn these words monomer and polymer. So remember that mono meant one. So that's one small piece that's going to repeat over and over and over again. But it's one piece. Mono means one, right? A monocle is one eye. A monogamous relationship is when they're one person and one person. That's a monogamous relationship. While we have a polymer, which is that one piece repeated many different times. Well, for this specific biomolecule, the nucleic acid, its monomer is called a nucleotide, which again, that should be a review from our biomolecules lesson. So the nucleotide has 3 parts to it: the phosphate group, the sugar, and the nitrogen base. Let's go get my DNA molecule. Okay, so we have this big DNA molecule, right? And you'll notice that there are essentially 3 pieces to each piece. We have the sugar phosphate backbone, which one of these is the sugar. That can be the phosphate. Actually, no, this would be, yeah, that's true. Sugar phosphate backbone, and then it's attached to a single nucleotide. So you see the white square, the black square, and one of these rungs on the ladder. That is a nucleotide. That repeats, right? So this entire thing is a polymer, and that one, 2, and 3, that little segment is called a nucleotide, right? It's one piece that repeats over and over and over again. This is what it looks like in 3D, and on your screen right here, you see it in 2D. Okay, so again, the monomer is a single nucleotide, so a single sugar phosphate and nitrogenous base, so a sugar phosphate and a base. That is a monomer. The polymer is many nucleotides linked up together to make a DNA molecule. So how does DNA actually give us our unique identity? So again, unique essentially just means what's different between 2 organisms. So between me and my mom, we have lots of things that are very similar between us, but of course each of us are different people. We have something that is unique between us. So what is exactly different between 2 organisms? Well, we have 2 options. It's either going to be the sugar phosphate, or it's going to be these bases that are in the middle. Well, the sugar phosphate never changes. It's always going to be sugar, then phosphate, then sugar, then phosphate, then sugar, then phosphate, then sugar, then phosphate, then sugar, phosphate. That is conserved between all organisms. The only thing that is different, that is unique, is going to be the order of these bases. Okay? Don't believe me? Well, let's break it down. So right here, the nitrogen bases. Again, we have 4 different ones. We have A and T, and then you have C and G, because apples go on trees and cars go in the garage. Those are the 4 different bases that can be there. The order of these bases is going to determine the sequence of DNA. So a DNA sequence, for example, could be ATCGAG. Another example of a DNA sequence could be TTTAC. Another example of a DNA sequence could be CCCCCCCCC. You know, it's just the sequence. The order of the bases determines the sequence of DNA. Okay, this sequence of DNA, as we showed in Central Dogma, is going to determine what amino acids, which are part of the protein, are made. So, for example, GAG in the DNA is going to code for glutamic acid. it's not making sense, that's okay. We're gonna keep going over it in different ways. So this right here is the nucleus of a cell. As you can see, we have the DNA inside of the cell, and the DNA is telling the nucleus, you should make some proteins. And the nucleus is like, oh wait, no, this is the nucleus. That is the whole cell. Sorry, I didn't notice this at first. Yes, so this is the nucleus, and the DNA is inside of the nucleus, and it's telling the cell, I think I'll make some proteins. But wait a second. We just said DNA is trapped and protected in the nucleus, and DNA codes for proteins. But proteins are made outside of the nucleus. Then how exactly can information from DNA get outside of the nucleus and into the protein factories if DNA cannot leave the nucleus? Well, let's talk about RNA. So we have this thing that is essentially a messenger. It's a messenger. It's literally called mRNA. That M stands for messenger. Its literal job is to be a messenger boy. Okay? So this is what it looks like in a cartoon form. So we have DNA is chilling inside the nucleus. DNA cannot leave because it's too important. It cannot risk leaving the nucleus. It cannot risk leaving its security. It cannot risk leaving its protection inside of the nucleus. So what DNA does is it just sends an errand boy, a messenger RNA. Okay? This is a messenger RNA. And the messenger RNA looks a little bit mad. He's like, I'm telling you, Carol, I'm done being this guy's messenger boy. He can leave the nucleus and do it for himself for all I care. Which obviously, the DNA can't do that, but it's just like a funny cartoon type thing, okay? So DNA, and we know this is DNA because it is double-stranded, and we know this is RNA because it is single-stranded. So, can DNA leave the nucleus? Yes or no? Why do you need to use RNA to send a message to the cytoplasm? So here's some hints. Where's the DNA? Where are proteins made? Are these the same place? Why do you need to use RNA? Good job. So, how exactly does DNA get copied into mRNA? Well, we're going to use that whole idea of complements once again. So, we know if this is a DNA strand and we're trying to find its DNA complement, then apples go on trees and cars go in the garage. However, RNA is single-stranded. And since it's single-stranded, it can't have thymine. For some reason, thymine only works for double-stranded DNA. So instead of having thymine, in RNA, it's going to have what's called uracil. It's going to have a U in it. So if I have a DNA strand and there's a G on one side, what goes on the other side? What goes in its complement? So if I have DNA, let me ask this in a good way. So if I have so basically this problem has given me a strand of DNA. We all can see that. How do we know it's DNA? It says DNA. Another way that we know that it's DNA, it has T's in it. DNA has T's. Okay? And it's asking me to find the RNA complement. So where there's a G, it's going to pair up with C. Where there's an A, it's going to pair up with, now you might think apples on trees, but again, we're asking about mRNA. mRNA does not have T's. mRNA has U's. So instead of writing a T, we're just going to write a U. Okay, let's go to the next one. There's an A. Normally we would think apples go on trees, but wait, again, we are looking at mRNA. We're not looking at DNA. DNA has T's, RNA has U's. Okay, let's go back to our DNA strand. Now we have a C. What goes on our complementary mRNA? G, cars in the garage. How about we have an A on our DNA? What goes on our RNA? A U. Okay, and if we have a T in our DNA, what goes on our RNA? Trees and apples. Okay, so again, one thing to note, mRNA does not have T's. Basically, if you're about to write a T and you're talking about mRNA, you're going to be wrong. You need to make sure you write a U. However, if there is a DNA that has a T in it and we're trying to make the complement mRNA, you put an A. Okay? It's a little bit confusing, but basically all you need to do is make sure in your mRNA strand, if it asks you for a complement of mRNA, there should be no T's in it. There should only be U's, A's, C's, and G's. Okay, if I ask you to do a DNA complement, then you will have T's because DNA has T's, RNA has U's. Okay, all right. Hopefully I explained that very clearly. If I didn't, we're going to do some practice and we'll catch any misconceptions. So we're going to practice finding the complementary mRNA sequence to this DNA sequence. So we already told this is DNA sequence. We also can tell this is DNA because we have T's. T's tell us this is DNA. If we had U's, we would be like, oh wait, this is mRNA. Okay, so let's do the first one together. So where there's a T on the DNA, what does it pair up with? A. Where there's an A on the DNA sequence, what does it pair up with? U. T pairs up with? Remember, talking about mRNA, it has U's. Okay. So you want to pause the video right now and check that. How do we know that this pink strand, how do we know this is RNA? Exactly. So we have these U's here. It also tells us that we're finding mRNA, so that's also another way that we know that's RNA. Ignore that. That's not supposed to be that. Okay, so again, DNA base pairing, apples on trees, cars in the garage. RNA base pairing is AU and cars in the garage. So C and G always go together. The only thing you have to remember is that RNA has U, DNA has T. Okay. So again, here's central dogma. So what we just talked about was converting from DNA to RNA. So we talked about DNA has AT-CG pairs, RNA has AU-CG, DNA is double-stranded, RNA is single-stranded, DNA must stay in the nucleus, RNA can leave the nucleus. DNA is the original source of all the message, RNA is just a copy of the message. Okay, that process of turning DNA into RNA is called transcription. Okay, so again, that's sending DNA's message out of the nucleus. Let's keep it moving. So RNA, there's actually 3 different types of RNA. We have what's called rRNA, ribosomal RNA. We won't really talk about that at this level of biology, but if you choose to study biology in college or even past college, you will learn about rRNA. What we talked about today is messenger RNA, that mRNA, which is the messenger that leaves the nucleus and brings DNA's message into the protein factories, which are the ribosomes. And then we're going to learn about transfer RNA in the next lesson. We're going to learn about transfer RNA. Yeah. So if you even forget like what mRNA is, M stands for messenger RNA. That should be pretty straightforward to remember. So how can you tell the difference between DNA and mRNA? True or false? mRNA is a copy of the DNA that is able to leave the nucleus. Another question, is mRNA required for a protein to be made? Okay, so you're going to take some time and watch this 2-minute video clip on the slides. And some things to keep in mind as you watch the video. Can the vaccine make you sick? Does the vaccine include the virus? What protocols are in place to ensure the vaccine is safe for public use? And how is mRNA used in the vaccination process for COVID-19? We just learned about DNA and mRNA's function. How is that actually being used in this? Okay, and the video again is 2 minutes long, so please watch it. We're not going to watch it right now. So now your actual question is, what is the role of mRNA in the fight against COVID-19? What is the spike protein, and how does a vaccine affect it? Okay, here's a little picture to try and help you. This is what the virus looks like, and this is the antibody. Again, we're not watching the video. I don't know why it keeps clicking. So essentially, the COVID-19 vaccine has mRNA that makes our body produce the spike protein. Okay, and if our body is producing the spike protein, our body is also going to naturally produce antibodies to fight that spike protein. That way, if you come into contact with COVID-19 somewhere, the body already knows, I've seen this spike protein, I know how to beat it, and the body can beat it. Okay, that's basically a summary of how this vaccine works. Okay, so let's talk about proteins. This is our last section. We're almost to the end of the home stretch. So we know DNA goes into mRNA, and then what? What's next in central dogma? What are those last couple things we need to know about central dogma? Well, after mRNA is made, it's going to leave the nucleus. That's its whole job. It's a messenger. And once it leaves the nucleus, it's going to find this thing called a ribosome. Look how cute this drawing is. I was like, this is so cute. So mRNA, which is represented by this yellow right here, is going to find a ribosome. Once at a ribosome, there are going to be 3 mRNA bases at a time. Those 3 bases are called a codon. That's going to attach to a tRNA molecule that has an amino acid attached to it. Okay? Those amino acids are going to connect together like this. And then once it's all the way done, it's going to fold up and make a protein. So again, the mRNA leaves the nucleus, it comes to the ribosome. 3 bases are read at one time. Those 3 bases code for 1 amino acid. Those amino acids are going to link together in a chain. They're going to fold all up, and then boom, they're a protein. They go and do whatever job they're supposed to do. Oh, is that? Okay, so here's another more simplified way of saying what I said. So this is the mRNA sequence, which I just caught an error on this picture. I just got it from Google. Does mRNA have T's in it? No, mRNA does not have T's in it. So this image has an error in it. Those are supposed to be U's. So whoever made this picture didn't know what they were doing, but that's okay. It makes for a good learning lesson in class. So a ribosome is going to read 3 bases at a time. Those 3 bases are again called a what? They're called a codon. 3 mRNA bases at a time are called a codon. They code for 1 amino acid. Okay. So again, the process of matching an amino acid with an mRNA to build a protein is going to be called translation. Each codon has a code that matches up with a specific amino acid. So again, ACC is going to code for a specific amino acid. While GTA would code for another amino acid. So every 3 bases codes for 1 amino acid. Okay, the piece of the tRNA that matches up with the codon, which is very creative of them. There's a codon and there's an anticodon. That makes it easy. Codon and anticodon. Okay, so again, ribosomes are going to be those protein factories. So there's ribosomes that are free floating in the cytoplasm. And they are just literally protein factories. That's their only job, is to make proteins. Okay? So that process that we just talked about was creating RNA, or turning RNA into proteins, turning RNA into amino acids, which then fold and make a fully functional grown-up protein. So translation is turning that message into an actual functioning protein. So why are proteins so important? Well, I mean, if you were to read this image, you can see we make your hair, nails, eyes, skin, blood, bone, muscles. We work for your transportation. We repair and replace tissues. We are basic building blocks. Antibodies are proteins. So yeah, they basically do everything in your body. Structural features, body processes, all of that. They're very important. Okay, so here's a fun fact. Well, not really fun fact, just something that it took me a minute to wrap my mind around this, but this is how it works. So plants make amino acids. They string them together to make proteins. When you digest a protein, your body's going to break it down. So let's say I take a spinach leaf, I bite it, I break it down. Inside of that spinach leaf, there's all these different proteins in it. So I'm going to break it down into its individual amino acids again. And then as I'm making my own proteins, then my cells are going to use those recycled amino acids back together in a new order. So the body is actually very efficient. There's lots of recycling that goes on, as why we have to eat food, and that's why you have to eat plants and whatnot, because they make all amino acids. We don't make all of them, but they plants by they, I mean plants. Plants make all of them, and humans cannot. Humans and other animals cannot. That's why it's very important to eat your vegetables. Okay, I'm starting to ramble. I'm sorry. But we're at the end of the lesson. So let's do our summary of the lesson. So for our summary, DNA must stay protected in the nucleus. That process is called transcription. Or DNA stays protected in the nucleus, and then DNA undergoes a process called transcription. Transcription converts DNA into RNA. RNA carries DNA's message from the nucleus out into the ribosomes, which again, ribosomes are protein factories. Um, RNA is translated into amino acid proteins, and those proteins actually control cell functions. Then those proteins undergo what's called gene expression, which we'll get to that much, much later in the year. They control your traits, so your organism's characteristics. So whether your hair is curly, whether you're tall, whether your eyes are blue, whatever it may be, your genes get expressed. So this is central dogma, the law of life, also known as protein synthesis, also known as both transcription and translation. So yeah. So so things that you should know is that DNA is double-stranded. You should know the basis of DNA. You should know that transcription is the process of turning DNA into RNA. RNA is single-stranded. There are no T's, there's only U's. And then they have this process of translation that turns RNA into a protein, a functioning protein. And you also need to know about RNAs that they are read in bases of 3. So every 3 bases codes for 1 amino acid. Every 3 bases codes for 1 amino acid. Okay? And don't worry if this isn't making 100% sense to you, because like I said, tomorrow we're going to go more in depth in what transcription and translation is. Today's just kind of this overview, setting you up for the rest of this unit and whatnot. We love DNA. We love biomolecules, and we will be doing a lot of work with them. So yeah,